Okay, this looks a little weird. When I imported it into my iPad, the text got messed up, but I think you can still read it. So next we're going to talk about capillaries. So it's important to realize that the function of the capillaries is, that's where, like I always say, the magic is going to occur. So that's where the materials are going to be exchanged between the interstitial fluid, so the fluid around the tissue, or within the tissue, and the blood. Okay, so it's when gas, like oxygen and carbon dioxide and glucose and everything, that's how everything is going to be exchanged. Okay, now the structure of capillaries. So capillaries, they only have an endothelial wall. Okay, so they just have this wall, which is on here, it's kind of this blue color. And then they have a basement membrane. Well, actually, okay, so this is the basement membrane, is the blue, okay, and then the endothelial wall is the pink. So this is the endothelium, and then there's the basement membrane. So basically, it's a whole bunch of simple squamous cells tightly packed together, and they're laying on a basement membrane. Okay, so the capillaries are super, super thin. Okay, so that makes sense when you put structure and function together, right? So structure and function, if you're really thin, it's going to be easy to get things through. So easy for gas exchange to take place, easy for nutrients to be exchanged. Okay, now there's 3 types of capillaries. So the first type are continuous. So continuous means that the epithelial cells, these squamous epithelial cells, are touching. Okay, so they're pretty close together, but it's not sealed. So things could go in between them, things could go through them, things can kind of go around them. Okay, so they're not sealed together. Okay, so this is the most common type of capillary, so it's found like everywhere. So muscle, skin, lungs, and the nervous system. Okay, so things can flow through by diffusion, okay, or we could have bulk flow, which would be like endocytosis, exocytosis. Okay, now the next type, whoops, what did I do? The next type, I want to get rid of that. The next type of capillary are called fenestrated, so they have holes in the cells. So the holes are pores or fenestrations. And so the epithelium is whole, okay, so you could look at this again, but in there are little holes. Okay, now, they're still going to keep blood cells in, so only small things can go through, but because it has those little holes, things are going to be able to move faster through it. Okay, so we're going to find those in places where we need rapid secretion and absorption, okay, where we need things to move fast. So they're going to be places like small intestines, the choroid plexus in your brain, where the cerebral spinal fluid is formed, in the kidneys, and the endocrine glands. Okay, so it's basically the same things moving through, but it can move faster because there are holes there. And the holes are called fenestrations. Okay, so the next thing are sinusoids. In sinusoids, they're modified capillaries. So the capillaries are not, they're irregularly shaped, and they have big holes in them. Okay, so if you look down at this picture, which is from the book, they have these big openings in them. So anything can go through those big holes. So anything can go through. So blood cells could go through, platelets, proteins. Okay, so if this is a blood vessel, okay, it might have a hole like that, so that the blood cells and the proteins and anything in there can leave. Okay, so this is how big molecules, so one is a protein called albumin, how they're going to get from where they're being formed into the blood. Okay, so this is how blood cells get in your blood, this is how albumin. So usually they say that organs that have sinusoids look red. Okay, so liver, spleen, bone marrow, and then some endocrine glands. Okay, so sinusoids. Okay, now, how do we control what's going on and how do we control how the blood is moving through the capillaries. Okay, so before we do that, we have to talk about two more parts that we haven't talked about. So here's an arteriole, okay, and then here's the venule. Okay, now those are connected by this thing. It's called the meta-arteriole. Okay, so maybe let me try to make this wider. Let's make it black. Okay, so the meta-arteriole is coming down and it's extending off the arteriole. Okay, now it actually, the name of it changes as it goes, and then they call it a thoroughfare channel as it gets closer to the venule. Okay, now coming off of this meta-arteriole are these things called precapillary sphincters. Okay, so what's a sphincter? A sphincter is a circular muscle that can close things off. Okay, so these little yellow spots, those are precapillary sphincters. So basically, the blood can flow onto this bypass. If the sphincters are open, okay, the blood will go into the capillaries. Okay, if the sphincters are closed, the blood doesn't flow through the capillaries. Okay, so that's how we're going to regulate it. Okay, so that's what I have written down here. Okay, so if the sphincters are open, the blood goes through the capillaries, okay, and the tissue gets blood. So the tissue is perfused, so it's oxygenated, okay, and the waste is taken away. Okay, if the sphincters are closed, the blood bypasses the capillaries, and then there's no exchange. Okay, so what's going to be important is in our body, there's not enough blood to go into every capillary bed that we have. So we're only going to give it to the tissue that needs it. Does that make sense? Okay, so here's what I just said. Okay, so it's important to realize that we don't have enough blood in our body to fill all of the capillaries at one time. That's an ugly little line there. Okay, so we don't have enough blood to fill all the capillaries. So we have about, they say, 60,000 miles of capillaries, but we only have 30 milliliters of blood in them. So what happens is we cycle it. So sometimes we have blood in capillaries and sometimes we don't. So the precapillary sphincters, they contract and relax, and that's called vasomotion. Okay, here's another vocabulary word that's important. Okay, so perfusion. Perfusion is the amount of blood entering the capillaries per unit time per gram of tissue. So it's measured in milliliters per minute per gram. So perfusion determines how much blood is going to the tissue. Okay, so you want your tissue to be perfused so that it has oxygen. If you don't, that's when you get ischemia and where you could get infarction. Okay, so perfusion. All right, now this is an animation, and I don't think it's going to work on here, but when you guys view this PowerPoint, you're going to be able to click on this little arrow, and this just shows you if precapillary sphincter is closed. Okay, so if these are arterioles and they're constricted, the blood doesn't flow through them. I hope that makes sense. If they're open, the blood will travel through them fastly. That's not a word. Quickly. It'll just flow. If they're closed, okay, so say these little things are arterioles and this is blood supply. If they're open, blood flows through. If they're closed, it doesn't. So, but you can see that animation if you look at this as PowerPoint slides. Okay, now, venules and veins. Okay, so we're at the capillaries and we want to get the blood back to the heart. Okay, so we're going to go through venules, which are little veins. Okay, the proximal venule really has a lot of holes, and it almost functions like a capillary. Okay, then we go to small, medium veins, and they have valves, and these will be what we name. Okay, and they prevent the valves prevent the blood from pooling. It keeps the blood flowing in one direction. Okay, the valves are actually made of the inner layer of the blood vessel wall, so the tunica intima, and they have elastic and collagen in them. So the valves kind of look like heart valves. Okay, so there's the valve. We want the blood to flow back to the heart, so the valve will close and the blood won't go back. It'll just keep going forward. Okay, now systemic veins are veins like the brachial vein, the femoral vein. Okay, so in general, veins have really low blood pressure. Okay, so the blood pressure in them is like 10 millimeter of mercury, and it just stays low. It's kind of like a lake where nothing is flowing, and arteries are more like a river. Okay, so veins have really thin walls. They have less smooth muscle and less elastic tissue. Okay, now what's special about them? Okay, they have valves for sure. Okay, but they expand really easily, and they have what they call high capacitance. So basically, capacitance means they have a high capacity, so they can hold a lot of blood. So they serve as, your book calls it a blood reservoir. So in your body, at any one time, most of the blood is hanging out in your veins because they're big and floppy. Okay, and that's normal. That's where it should be. Okay, so here's a picture that represents that. Here's the distribution of blood at rest. Okay, so 18% of the blood is in the pulmonary circuit, so going to your lungs. 12% is going to your heart. 11 is in the arteries. 5 is in the capillaries, and then 54% of the blood when you're at rest is in your veins. Okay, now if you start exercising, these change a little bit, right? More will go here, here, and here, and this will drop. But your veins are a reservoir for blood. Okay, now I am going to stop right here and upload it, and then we'll restart here.